The 2018 Nobel Prize in Chemistry recognized the transformative power of harnessing evolution's engine for human benefit, specifically honoring Francis Arnold for directed evolution of enzymes and George Smith and Gregory Winter for phage, display of peptides and antibodies. This award acknowledged innovations that have revolutionized fields ranging from industrial catalysis to pharmaceutical development, offering potent tools to create novel biomolecules with tailored properties. These methodologies transcend mere imitation of nature. They represent a profound ability to guide and accelerate evolutionary processes to solve specific problems. The concept of directed evolution emerged from a growing appreciation for the limitations of rational design in protein engineering. Traditional methods, relying on detailed knowledge of protein structure and mechanism, frequently struggled to produce enzymes with desired characteristics. The unpredictable complexity of protein folding, coupled with intricate relationships between structure and function, often stymied attempts to engineer improved catalysts through purely computational or structural approaches. Instead, Arnold championed a paradigm shift, embracing randomness and selection as guiding principles. She drew inspiration from the natural evolutionary processes that have sculpted enzymes and other biomolecules over millennia. Arnold's pioneering work on directed evolution involved iterative rounds of mutagenesis, selection, and amplification. A gene encoding an enzyme of established activity undergoes random mutations, generating a diverse library of variants. This library introduces a spectrum of modifications, some deleterious, some neutral, and a precious few that might enhance the desired function. The key lay in devising efficient screening methods to identify these rare, improved variants. Arnold innovated selection strategies tailored to specific catalytic properties, allowing her to isolate superior enzymes from vast populations. These improved variants served as templates for subsequent rounds of mutagenesis, further refining the enzyme's performance through successive generations of evolution. This iterative process, mimicking natural selection in a laboratory setting, allowed Arnold to breed enzymes with enhanced activity, stability, or substrate specificity. One notable early success involved engineering subtilisin E, a bacterial protease, to function effectively in dimethyl-formamide, an organic solvent inhospitable to most enzymes. This achievement showcased the potential of directed evolution to create biocatalysts capable of operating under harsh conditions, expanding their applicability in chemical synthesis. The traditional approach to enzyme engineering would begin by painstakingly studying the existing subtilisin E structure and making educated guesses about modifications that might lead to dimethyl-formamide tolerance. Arnold's approach bypassed this laborious process, instead letting evolution unveil unexpected solutions not readily apparent through rational design. The resulting enzymes, evolved through several generations of mutagenesis and selection, displayed remarkable activity and stability in the organic solvent, demonstrating the power of directed evolution to overcome limitations inherent in rational engineering. The impact of directed evolution extends far beyond the initial demonstration with subtilisin E. It has since become a widely used technique for improving a diverse range of enzymes, impacting industries such as biofuels, pharmaceuticals, and materials science. Directed evolution has enabled the creation of enzymes that can efficiently degrade cellulose for biofuel production, synthesize chiral drug intermediates with high enantiomeric excess, and catalyze the formation of novel polymers. Its versatility stems from its ability to adapt to diverse selection pressures, allowing for the optimization of enzymes for nearly any desired characteristic. The method has influenced the construction of enzymes tailored to function at elevated temperatures, withstand extreme pH levels, or utilize unnatural substrates. In essence, directed evolution offers a powerful platform to customize enzymes for specific industrial or therapeutic applications. Parallel to Arnold's work on enzyme evolution, George Smith and Gregory Winter independently developed phage display, a complementary technology for evolving peptides and antibodies. Phage display utilizes bacteriophages, viruses that infect bacteria, to present diverse libraries of peptides or antibodies on their surface. This technique provides a physical link between the displayed protein and the genetic material encoding it, enabling efficient selection of proteins with desired binding properties. Smith's initial contribution involved developing the fundamental technique of displaying foreign peptides on the surface of filamentous bacteriophages. He demonstrated the possibility of inserting foreign DNA sequences encoding short peptides into the phage genome, causing the phage to express these peptides as fusion proteins on its coat proteins. This breakthrough laid the foundation for creating vast libraries of peptides, each displayed on a separate phage particle. The crucial facet of Smith's approach was the physical linkage between the phenotype that is, the displayed peptide, and the genotype that is, the DNA encoding that peptide residing within the phage particle. This linkage enabled the subsequent selection and enrichment of phages, displaying peptides with specific binding affinities. Winter further refined phage display, focusing on the evolution of antibodies. Antibodies, essential components of the immune system, recognize and bind to specific antigens with exquisite precision. Winter recognized the potential of phage display to generate human antibodies with therapeutic potential. He engineered phage libraries displaying antibody fragments, specifically the variable regions responsible for antigen binding. By panning these libraries against target antigens, Winter and his colleagues isolated antibodies with high affinity and specificity. This approach circumvented the need for traditional hybridoma technology, which involves immunizing animals to generate antibodies, often resulting in immune responses against animal antibodies in human patients. A landmark achievement was the development of Humira, adalimumab, a fully human antibody approved for the treatment of rheumatoid arthritis and other autoimmune diseases. Humira was generated using phage display, demonstrating the clinical potential of this technology. Since Humira's approval, numerous other antibody therapeutics developed using phage display have entered the market, revolutionizing the treatment of cancer, infectious diseases, and inflammatory disorders. Phage display continues to be a valuable tool for antibody discovery, enabling the generation of antibodies with tailored properties, such as increased affinity, improved stability, or novel binding specificities. The technique has also found applications in areas beyond antibody engineering, including the identification of peptide ligands for drug delivery, the development of diagnostic tools, and the study of protein-protein interactions. The impact of directed evolution and phage display reaches far beyond the confines of academic laboratories. These technologies have spawned numerous biotechnology companies, driving innovation and economic growth. The ability to engineer proteins with tailored properties has created new opportunities in diverse sectors, impacting industries ranging from healthcare to agriculture. Companies are using directed evolution to develop sustainable manufacturing processes, create novel biofuels, and engineer crops with enhanced nutritional value. Phage display has become an indispensable tool for antibody discovery, fueling the development of new therapies targeting a wide range of diseases. The development of directed evolution and phage display did face skepticism early on. The idea of relying on random mutagenesis to improve complex biological systems seemed counterintuitive to some scientists who believed in the power of rational design. Some expressed concern that the resulting enzymes or antibodies might lack the stability or specificity required for practical applications. However, the resounding success of these technologies, demonstrated through numerous examples of improved enzymes and clinically effective antibodies, silenced the critics. The methods proved robust, adaptable, and capable of generating solutions to complex problems that exceeded the capabilities of purely rational approaches. A crucial aspect of both directed evolution and phage display lies in the design of effective selection or screening methods. The power of these techniques hinges on the ability to efficiently identify the rare variants with desired properties from vast libraries of diverse molecules. The development of high-throughput screening assays, coupled with sophisticated selection strategies, has been instrumental in the success of these technologies. The selection pressure exerted during the evolutionary process dictates the properties of the resulting molecules. Careful consideration of the selection conditions is essential for optimizing performance for a specific application. The 2018 Nobel Prize in Chemistry recognized a paradigm shift in our approach to manipulating biological systems. The laureates moved beyond attempts to rationally design complex molecules, embracing the power of evolution to solve problems that were previously intractable. These technologies continue to evolve, with ongoing research focused on developing more efficient mutagenesis methods, improving selection strategies, and expanding the range of applications. The future promises even greater advances in enzyme engineering, antibody discovery, and the creation of novel biomolecules with tailored properties. The potential of these technologies to address global challenges in healthcare, energy, and materials science remains immense. In conclusion, Francis Arnold, George Smith, and Gregory Winter's pioneering work on directed evolution and phage display has revolutionized the fields of chemistry and biotechnology. Their innovations have provided powerful tools for engineering enzymes, peptides, and antibodies with tailored properties, impacting industries ranging from pharmaceuticals to biofuels. These technologies represent a profound ability to harness the power of evolution for human benefit, offering solutions to complex problems that were previously beyond reach. The legacy of these laureates will continue to inspire scientists and engineers to explore the vast potential of biological systems for innovation and discovery. Their work stands as a testament to the transformative power of combining scientific insight with the elegant simplicity of natural selection,